आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़।
Celebrating Azadi Ka Amrit Mahotsav, we bring you a podcast series on STEM popularization, science, technology, engineering, mathematics and medicine, supported and catalyzed by the National Council for Science and Technology Communication, Government of India. This program is produced by Media for Community Foundation. Creative content and technical support is given by Purvasha Banerjee and Shushanto Das.
एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़, की आवाज़।
Indian Statistical Institute is a public university which is recognized as an institute of national importance. It grew out of the statistical laboratory set up by Prashanta Chandra Mohananobish in Presidency College, Kolkata. Established in 1931, this unique institution of India is one of the oldest institutions focused on statistics. Primary activities of ISI are research and training of statistics, development of theoretical statistics and its applications in various natural and social sciences. We welcome Dr. Raghunath Chatterjee, who is a professor and head of the Human Genetics Unit at Indian Statistical Institute, Kolkata. In this podcast, Sir talks about the microRNA and its role in human health and living.
Myself, Raghunath Chatterjee, currently professor and the head of the Human Genetics Unit, as you mentioned. So I did my master's in physics, and then I started working. My PhD was in biological science. Mainly I focused on the genetics, genomics. And after completion of my PhD, I went to USA. I worked at the National Cancer Institute, USA, and I worked mainly on the epigenetics over there and transcription biology and how the gene regulation is actually governed by the epigenetic mechanisms. So after staying over there for around 5 years, so I came back to India and joined Indian Statistical Institute Human Genetics Unit. So I'm primarily working with genomics and epigenomics in human health and diseases. So currently we are focusing on 2 diseases. One is psoriasis, which is a skin disease, and another is oral cancer. And most of you know the oral cancer, which is one of the most prevalent disease among the men in India. I'll talk about a specific type of molecule, so that is called the microRNA. So these are small non-coding RNA. So normally you know that messenger RNA, they code for protein, and proteins are now functionally relevant. So they do a lot of functions in the cell. But there are a lot of non-coding RNA. So one of them is a small non-coding RNA that is called the microRNA. And microRNA has a very specific role. So the microRNA actually bind to the untranslated region, 3' untranslated region of the mRNA, and they control the gene regulation. So once they bind to the UTR region, untranslated region of the mRNA, and then they inhibit the expression of the gene or the post-transcriptional, so that is essentially the protein product is not formed from that mRNA. So it essentially regulates the gene expression. And because of this particular feature of the microRNA, so people actually tried to use whether the microRNA can be used to different, because in cancer we have seen that a lot of genes which are actually getting upregulated, so these are called the oncogene, and a lot of genes which are getting downregulated, these are called the tumor suppressor gene. And sometimes if you target some set of oncogenes which actually due to their suppression, the the disease can be controlled, or somehow, the progression of the disease can be stalled. So because of that, people started using microRNA as a therapeutics, and I also actually started working in that area. As I said, that the microRNA target a particular gene, so it binds to the untranslated region and then do a downregulation of the gene. But there is a problem there. The problem is one microRNA can actually target multiple genes. So now we see a problem, the problem in a very layman language, that let's say that microRNA 1, okay, so just a name, okay, it's not the real name. So let's say microRNA 1. MicroRNA 1 can target, let's say, gene A, B, C and D. But gene A and B, these are the two oncogenes which you want to actually downregulate it, but you don't want to disturb gene C and D, because these may be the housekeeping gene. They are actually required for the cell. Now, and if gene C and D are downregulated, then there might be some cytotoxic effect and some other side effect. So your intention is to reduce the gene A and B. Now, if you target, use microRNA A, send it to the cell, then it is not only going to downregulate gene B, it is going to downregulate gene A, B, C, D, all four. But because of the CD, there are a lot of cytotoxic effect. And because of that, a lot of microRNA which actually went through clinical trials, some of the microRNA which actually passed the clinical trial, some phases, but it was stopped because of the cytotoxic effect. So what I thought, that instead of taking one microRNA, if we take two different microRNA, so we took a very different strategy, but I'm not going to discuss the whole details of the research, just giving a very brief overview of the research. That let's say there is another microRNA, microRNA 2, which is also in the disease condition, both microRNA 1 and 2, they're downregulated. So now the microRNA 2 is targeting gene A, B, E and F. So now instead of taking one or two separately, if you take both of them together with very low concentration, and if there is a synergistic effect, then because of the low concentration of both gene microRNA 1 and 2, it's going to target gene A and B. But as you are using a low concentration, so gene C, D and EF, they are not going to be affected that much. So basically you are essentially making the target very specific. So that was the approach we started with. But there are a lot of other questions, like if the same microRNA is targeting gene A and B, then their binding sites in the UTR region, how far they should be, whether there is any other specific biochemical mechanisms which can actually dictate that what concentration you should use. So all those different kind of theirs, and we use a lot of fancy molecular biology as well as advanced different kind of genomic technologies, and we come up with again that a specific concentration of the microRNA can be used to specifically target the genes. And we use these particular thing in some of the cancers. We worked on oral cancer, we worked on mainly the cell line model system we have used, and also we worked on hepatocellular carcinoma, prostate cancer. So different kind of cancer model cell lines we actually used, and we found that this particular strategy is perfectly working. So this is one part of the work, which is a very important therapeutic outcome which may have. But again, as these are essentially the concept, the proof of concept we started with, we proved the proof of concept is working, but there are a lot of other things we need to do. For example, we worked on the cell model system, so cell line system, but in cancer it's not only a certain type of cells, homogeneous population cells are there. So it's a tissue. There are a lot of different kind of cell types are there. So a lot of heterogeneity are there. So we need to test in the tissue. So we actually do not have a mouse model system. So instead of that, what we are currently doing is essentially we are doing a 3D culture, spheroid culture, and with that actually we are using this set of microRNA and trying to see whether with these we can actually reduce the cancer progression. So that is another thing. But once it is done, then we need to actually go beyond that. We have to use a mouse model system and then whole system before actually it can be transferred to any kind of industry for any translation, to see the translation component. So this is where we are currently now.
We thank Professor Chatterjee for being with us in this podcast episode and sharing his knowledge and experience.